I think maybe the floor is mine then to ask you some questions. Before I do so, I wanted to say congratulations. I found it an incredibly lovely thesis to read. I think you cited lots and lots of relevant literature. You must have been very busy reading. You've clearly done some exciting field work, amazing data set, thanks to your supervisors as well, of course. And I also found the thesis really beautifully illustrated, as we can also see here. So congratulations on a job really well done. And I thought, I mean, I have some nitty-gritty questions on some of the chapters too, but I thought maybe we can start out a little bit more generally. And I wanted to ask you how much you think there should be behavioral buffering towards reproductive success in your study species.
That is a very relevant, yeah, obviously it's a very relevant question, but, and one I wondered a lot. So when we look at, maybe I can go up in the slides. Yeah, okay, it's lagging a tiny bit. Where is it? It's here. Yeah, when we look at this, I mean, obviously you've read it in the thesis, but yeah, I was wondering, because when I first saw the data before fitting the curve, I thought, okay, and this is explained as is in the thesis that we did it a posteriori, that I was wondering whether there would be a plateau here, so an indicator of the foraging effort, the behavioral metric, increasing without effect on the reproductive success. Well, this is, as you can see here, it's maybe tough to believe from this figure, but when I try to fit a breakpoint regression to this, it indicated like a possible breakpoint around 30 hours of foraging trip duration. So yeah, I think it's possible. I think with less messy data, maybe that would show a bit more. Yeah, but so in penguins, that's also something we discussed a lot with the collaborators, because in ALCIDs, for example, this has been shown. I have maybe, okay, yeah. I just prepared, because I think it's really important. I kind of knew that we were going to talk about this, because it's again a figure I put in the manuscript. But like the inspiration I took when I did that was looking at this figure on common years in the UK, and in this case this is very clear, the behavioral buffer before the vital rate is affected. And we have also evidence, this is also a paper by Leighton Matthews of this on the body mass of geese, well, not seabirds, but not so far. But yeah, in penguins, so I believe there may be something, because especially because I mean, why could they, in Adelie Land, they almost never, the mean foraging trip duration almost never, I mean, never drops below 16, 15 hours. In the Antarctic Peninsula, they are able to do trips in 9 hours, less than 10. So even though the buffer may not be as huge as the one the ALCIDs have, I think, yeah, penguins have some.
Yeah, so related to that, so you see that the trip duration responds to something, and then it is related to reproductive success. How about it being related to adult survival? So what do these long trips bring for adult survival?
Yeah, that's one of the metric we thought using, actually, because yeah, the behavioral or physiological component of the timeline is missing for adult survival, and the very metrics I really wanted to use at the beginning were metrics from the Weybridge system, because I thought they would be more informative than the metrics just from the duration of the trips. But as I couldn't crack, I mean, I think it's explained a bit in the thesis, I couldn't crack it far enough to have enough data to make this relevant. So, but yeah, that's something I'd like to use. But I'll be surprised. This actually links a bit to a work I did before the thesis with Kyle Elliott in Canada, working on pelagic cormorants on Middleton Island. And what we found is that the energy expenditure, either at the individual level or at the population level, wasn't related to survival cost. And I think the conclusion of that was that I thought was maybe it's just their condition, actually, that at the end of the breeding season that will determine their fate. And if we have only the measure of the effort they do, we may not be able to see the impact on survival. It's really have to combine the effort with the condition it leaves them at the end of the breeding season.
And so would this also have to do with individual variation in quality behind your population level estimates?
Yeah, that's also the big question. Well, that's why I think also a strength, once we can crack the code of the mass tracker, a strength of this this setup is that we can have an idea of how low during their life individuals can go in terms of body mass. So in this way we would have a mean to scale the condition of an individual compared to what's its usual condition, and that would be a way to account for possible differences, because it's really possible. I mean, the idea of quality is that, applied to this case, is that there may be individuals that are 4 kilos, both of them, and one goes at 3.5 survives, and one goes at 3.5 and dies because of this decrease. So, but we would be able to control for that by looking at the longer time series, I think the longer picture with all the mass variation across years.
So maybe moving in a slightly different direction, you looked at adult survival and juvenile survival, and I was wondering whether you think that there are different drivers for these. So obviously you looked at the penguin relevant sea ice measures, and I must admit I'm not very familiar with sea ice measures, so I got a little bit confused about the different ones that you have used and how they do or do not connect to each other.
Yeah, well, most of the metric used are sea ice concentration, so really an area, usually hundreds of kilometers, and it's the percentage of this area covered by sea ice. The thing is, I think the big question you're asking is whether there is spatial, to some extent, but mostly temporal autocorrelation in the sea ice. Because the time frames at which sea ice influences juvenile survival and adult survival may be quite different, actually. So they may be connected if the sea ice, for example, in April, is also important in the winter where we have more evidence that it's affecting adult survival. But yeah, for this, I don't know. I know it's a point that I have to investigate more. Is how much, because it also depends on the scale, right? The temporal and also spatial scale, this autocorrelation. So yeah, and yeah. So yeah, I think it's a complete answer. Or if you're not convinced, I can try and develop more.
Yeah, so I was also wondering about these lag effects. You mentioned them briefly, but for example, these 2014 and 17 cohorts, the juvenile survival you of course take into account across multiple years. So whether these juveniles did or did not experience these two massive events, was that something that you took into account? Do you think that would also only matter for the breeding adults, or do you think it would also affect the juvenile survival?
Yeah, that's yeah. I don't think it would affect the juvenile, because let's take the first extreme event, 2014. So the juvenile is born in 2013, or let's say 2012. 2014, the year of the extreme event, should have been the year where they came back to the colony, to visit the colony. But the juveniles not being attached to the breeding site, and especially the first year where they come back, they don't come back as early as the adults. They usually come back in January, so quite at the end of the reproductive season. So I think also because they are not attached to the site, they may not, like if maybe they perceive that they have to cross too much sea ice, they wouldn't take this risk as the adult would do. So I think it plays on the detectability of the juveniles, this extreme event, so they may not be able to come back to be detected by our antennas in those years, but that's why capture-recapture models are for. But I don't think it may affect them this much.
I was also wondering about the predictive power of these adults. So could they not see it coming in 2017, that they needed to cross this exceptionally long stretch of ice before getting to the water and give up their breeding attempts a little bit earlier? I mean, they ended up with 0 reproductive success anyway. Could they not have saved themselves in the process?
Yeah, intermittent breeding, but I wonder, I mean, for other long-lived species, it's like to skip breeding is a really common strategy, and we have some suggestions that they do that at the southernmost colonies in the Ross Sea, but in Adeliland, looking at the data, I haven't tested it properly, but just looking at a lot of data, the breeding cycle we could see with the RFID, we can have it with all the years, They just don't seem to skip. Our mature birds, I never see them skipping. So I wonder, I mean, that's, I think, one of the suggestions, that there is different level of skipping depending on the latitude. But yeah, I don't think in Adelaide they have this adaptation of skipping reproduction. They have do it all the time.
But even
strategy.
without skipping, would they not notice during incubation, for example, that things weren't progressing as much as they were maybe expecting or hoping for, and therefore giving up during incubation already?
Okay, now I get your question. I think it really relates to the distance they had to walk, because there has been a lot of work in physiology. So before doing this kind of life history stuff at Point Geology, they used to do, the researchers there, they did a lot of physiology work, and notably they worked on the how is it called in English? When they stop eating.
Fasting.
Yeah, fasting. They did a lot of work on fasting and on the phases of fasting. And so they could show that there is a trigger that is hormonal, and also a trigger of behavior, of body condition that triggers reproductive abandonment. But actually this trigger, the threshold at which this is triggered, it's quite likely that it has been shaped by natural selection over a long period. And what I would suspect is that those extreme events, they must not have happened a lot of time in this evolutive history, so that the threshold at which reproductive abandonment occurs doesn't allow the individual to walk over maybe more than 50, 60 kilometers of sea ice before being able to replenish the reserves. That's what I agree, maybe that wasn't super clear when I presented it, but that's the idea.
So could you bring these figures back up of the places that they needed to cross to get to the open water in these 2 years? Because the direction of travel seems to be different in these 2 years as well. So, you know, you've drawn the arrow to where the nearest open water is. Do they know this? Is this repeatable across years? Do they prefer to move in one direction or another? Is there any navigational work on penguins?
I must first apologize, because these are not tracks. These are me who drawn the tracks to the nearest. I should have precised this more. This is the distance to the nearest open water. But that is a very relevant question, and the answer is I don't know if they prefer. Definitely, when you look at the GPS data, when there is no fast ice, for example, they have preferences. They often go to the east, for example. But when there is ice like that, I don't know if, like petrels, they have the ability to sense through smell the nearest open water, but that is a good question. I think that the Japanese, they did a bit of experiment where they translocated penguins and measured how much time it took them and whether they traveled in a straight line to the colony. I think to some extent they must be able to do it, especially since when they come back for breeding at the beginning of before incubation, it's not unusual that there is let's say 60, 100 kilometers of fast ice, and when you are 100 kilometers away from the archipelago, you don't see it. So they must have some navigational tools that they can use to do that. So I would suppose that if they can do it in one way, probably they can do it in another one. But then it would be super interesting if only we had more data on this to look whether the individual with the best navigational abilities were also the one to escape, to be able to better escape this kind of trap. Yeah.
And I was also wondering, do you ever observe mortality? So if, for example, in 2017 they were actually succumbing to having to walk so far over the ice, would you be able to, I don't know, with drone flights or whatever, see dead penguins lying before getting to this water body to forage and replenish their energy?
That's a question I actually asked, because I wasn't there, unfortunately, in those years, and I asked to Thierry Raclot, which is a researcher in Strasbourg that was there, and he told me that he wouldn't see many dead individuals at the colonies, but what he suspected is that, I don't know if he observed that really, that a lot of them would fall into crevices on the fast ice. So I suppose that may also be a reason for the decreased survival in that year. But yeah, your question really highlights the fact that for adults, what would be ideal is to have an idea of if and where there is a peak in mortality, even not especially in extreme years, but in general. And this we don't really know. I mean, we suspect it's during the molt, because it's another energetically intense period right after reproduction, which is also intense, but we don't really know.
Are there any type of loggers that you can use to figure out how this is happening, where this is happening, and when?
Yeah, that's for the juveniles, the paper by Hink et al. In 2020, they deployed loggers, and they looked so Argos loggers that would send the data, and they looked what was the main period where the loggers stopped working, so indicative of probably the individual death. We could probably do something like that for adults, but the problem is that we would, you attach the logger on the feathers, and so you would attach them to post-breeding individuals that would only keep it for a month before they molt, and then you would have to attach them again. So a bit cumbersome, but maybe possible. And that's, at the moment, unfortunately, that's the only way I see to do it. Because I tried to talk about with people that did cruises in Antarctica to see where they would observe the adults in winter, and we have a bit of data that shows that they are associated with areas of heavy ice concentrations. But yeah, apart from that, to try and see where they die, I think it's quite difficult. But I'm sorry?
Yeah, we have to retrieve them. You were talking about.
Yeah, because we retrieve only the one that survived, so that's also the thing.
So maybe I'm now doing the chapters upside down, but I just wanted to ask you about this last chapter on the adult survival, whether you've also tried fitting a covariate for how much distance they would need to cross as an alternative to your sea ice measurement.
Yeah, that's right. I didn't do that, but I don't know if there are enough years with fast ice that they have to cross, but I mean, if your question was at the population level, I think there are not enough years, but one of the things I started working on and then I stopped to write the thesis is at the individual level, because, and I think that was also a question from you or from Richard, but from the RFID system we can have the date at which the individual is last seen, and there is a bit of variability in this date. Or maybe I put it in the thesis, I don't remember. Okay, I put it. And yeah, so at the individual level, I would suspect that the individual that have, when they exceed the colony, that have the longest distance to travel, they would be the one showing lowest survival compared to the one that have a shorter distance, assuming that they all leave the colony at the same body condition relative to their abilities / quality.
I think I have a few more minutes, right? Okay, so maybe I'm going to go now back to chapter 1 and ask a few more detailed questions on mostly the statistics actually for that one. So maybe starting with the data selection actually. So first you say that you focus on this chick stage because this is where the main or reproductive success differences get made, but you haven't actually quantified it, I think, in your thesis. So how, in terms of percentages, what is this peak nest failure? Are we talking about 60%, 90%, or?
Yeah, I don't remember the numbers. It's definitely, I mean, the studies that I cite on this is quite old. I've read more recent studies comparing the survival between the crèche stage and the guard stage, and we can see very clearly that the survival, I think, in the crèche stage is in excess of 90%, so really high in crèche. But I think your question is more about incubation also.
Yeah, it's more about the pre-hatching stage, because that would be... So what we see in the terns, for example, is that if they get in trouble, the sooner they quit, the higher their individual survival to the next season is. So it really benefits to know when you are winning and when you aren't winning. So I would think, I mean, it comes back to one of the previous questions a little bit too. It's like, can't they predict it during incubation already? So, and if you are not taking that into account, are you bringing a bias to your data set? Are you missing an important part? So the percentage of nest failure would be relevant for me in that respect. Like if it's 10%, maybe it's not so important, but if it's 30 or 40%, maybe then that would be important to take into account. And is there any reason actually from your data to not be analyzing it and having stage of reproduction in as a fixed effect, as a class variable in your models?
Well, it's also that the foraging constraints in guard stage versus incubation are not really the same. They are much less constrained, I would say, in incubation, because they don't have to bring food this much. I mean, in incubation, they are constrained only by the partner ability to keep, I forgot the term again, fasting. Whereas in the guard stage, they are constrained by the need of the chicks. But yeah, we could include it. Another reason, it's more of a practical reason, is usually we don't miss the trips with the systems, but in the incubation, when the system is set up too late in the season, so it's the overwinterers that stay there for the winter that put it in place, so at the end of September, beginning of October, and sometimes there is a bit of lag, and the birds are already in the colony when they set it up, so we miss a bit of information there. And so that was also a reason to focus on the guard stage, because it's the one for which we have the clearest information. But I agree with you, and actually that's something I try to discuss a bit, but also I think that these two stages may also be linked, so it's not a direct causal relationship that we see the relationship between guard stage trip duration and reproductive success, because we have also evidence from other colonies that in the guard stage, birds that have a lower body condition that they may have inherited from constraining for more constraints during incubation, they have longer foraging trips in guard stage because of their lower body condition that they may have acquired in incubation. So this is what, the way I figured it is that, okay, guard stage is also integrative of this, so it may not be so bad.
Yeah, so maybe related to this is that in your models you wrote somewhere that you're not accounting for repeated measures for your foraging, for your trip duration, but your reproductive success data, of course, includes both the successful ones and the non-successful ones. So I was wondering whether you wouldn't want to actually include that as a fixed effect and take into account the repeated measures that you have. What was the reason for not doing it? You have the data, so why are you not taking that into account?
So your question would be to look at the relationship between trip duration and reproductive success, but also at the individual level.
Yeah, or to, at the very least, at the population level, take into account the fact that you will have more trips included for successful breeders than non-successful breeders, and to kind of correct for that.
Yeah, I agree with you that would be, yeah, now you put it like this, that would be indeed something I should have done.
Well, you haven't submitted it yet, you can still do it.
Yeah, I will, thank you. And yeah, it's really, so the setup is super great, but something we try to do, I didn't talk much about this, it was a Téo BARRACHO thesis. We can assess, we can measure or more like see the sex of an individual or its breeding success just looking at the RFID patterns. And what we tried to do with Gael is to identify the date of the breeding failure by having ground truth data of birds we knew when they failed and RFID data, but it doesn't work actually. So that's a reason also I didn't use many data of failed breeders, because they had to be in the subset of nest that we were visually monitoring so that we had the nest failure date. And we cannot automatically yet predict it from the RFID patterns.
So maybe one last very general question from me. So you mentioned that, we now want to predict into the future, but also we want to act. And you've mentioned that in your thesis as well. And I was wondering whether you have anything specific on your mind, because this sea ice cycle seems a little bit ungraspable to me. So what can we actually do once we know what the consequences of it would be?
Yeah, well, as you say, for stopping the sea ice declines, there is not much to do. I mean, there is not much at our level, but the idea, I think it's a lot of times the same idea. What we can act on is reducing the amount, the diversity of stressors the population are exposed to. And in this case, one of the stressors that is actually increasing at the moment after a period of decrease is the fishery for krill in Antarctica. So the Soviets used to, and maybe other nations used to fish for krill in East Antarctica. They stopped after the collapse of the Soviet Union, and now it's increasing again. Also, the Chinese fleet is increasing. So that's one way we can act. But I agree with you that for the CIS forecast, at least for the next decades, there is not much to do. But not much to do, but also, like, that's also the idea of looking at several populations. So I'm not going to go too far and say that we could translocate birds from populations that are kind of more adapted or more able to respond to low sea ice, but at least that gives us an idea of how they will be able to deal with it.
Thank you very much.
Thanks to you.
Thank you. So now over to me again. Yeah, well, I mean, just want to echo what Sandra said. It was an excellent thesis. It was a pleasure to read and a great talk. Okay, so I'm just also going to switch from general to specific questions, just depending on my whim. The first one, and I think you allude to this in the slide that you showed up at the end, how much do you think that, or how much evidence, convincing evidence is there that morphological traits should show a response to deteriorating environment ahead of a vital rate? I mean, I know there's the paper on albatrosses that apparently concludes that wing length has shown a response to environment, but something like wing length and body size, there's a whole load of other selection pressures that, and also just pure physics and enzymatic kinetics that would suggest that wing length should be maintained, except when an animal is starving. So, and I would think that would also apply to most seabirds. So as you suggest, you might see responses in some of the vital rates before a morphological change. And I think that kind of brings me on to the question about how much do you think you can generalize across species, and particularly, I suppose, from the terrestrial to the marine environment and from more, R-selected to more K-selected species?
Indeed. I think it really depends on whether or not it brings us back to the first question of Sandra about the buffers, the behavioral buffers. And that is something I was thinking a lot for a possible postdoc, for example, is to try and meta-analyze if we could bring together this kind of relationship between let's say a behavioral more than physiological, because I agree with you, size is maybe too tricky for this. A behavioral trait and a vital rate, let's say a reproductive success, because it's the easy one. And if we could bring together data on this for many species and just to see whether a threshold, a nonlinear relationship exists, that would be a good indication that there can be a change in physiological / behavioral trait before the vital rate. And this echoes another meta-analysis from Curry, you likely know about it, but it just skipped this trait aspect, and it just linked the abundance of fish, or forage more generally, and reproductive success, and it showed that across several oceans, several populations, several species, there was this nonlinear relationship where the food abundance decreases for a little while before the reproductive success decreases. And I would guess that behind this, I wouldn't be surprised as if it was like on the common years, that it was the behavior that was compensating in the meantime. So I think the applicability of the timeline really depends on that, and also I try to highlight it, but on the how much ramping the stressors are, because if the stressors are pulsed, as in the case of extreme sea ice events, then it's not adapted for this kind of disturbances.
Yeah, thank you, that's great. So I've got another question. I'll stick with the life histories. Why don't Adelie penguins skip breeding sometimes? Is it because they have no real idea what the rest of the season is going to be like, and they are effectively pre-programmed? But if that's the case, why is that not the same situation elsewhere when they do skip?
Well, for skipped breeding to evolve, it has to be, I would say, given the different species in which it is present, I would say it cannot Like it cannot have appeared randomly, so there must be selective pressures for it to happen. So I would say in the case of Adelie penguins, because the sea ice can break up, maybe this is really tough to predict actually during the season when it's going to break up. So if they arrive, there is a lot of ice, they may, well, it's a bit of anthropomorphism, but they may assess the situation and say, okay, it's bad, but probably it's going to break up and then end up in this situation. So yeah, more often than not, I suppose that it breaks up and frees them. Well, that's
yeah, no, I'm sure you're right. I was also along the same lines. Is there any evidence that they changed the timing of breeding, or is that also relatively fixed? Because that might suggest there was more flexibility.
They are actually quite flexible on their phenology, at least their arrival phenology. That's one of the things I looked at a bit at the beginning of the thesis, and you had, like, we thought about writing a paper on that with Céline and Nico. You had 15 days advance in their phenology at Point Geologie. I think it was linked to this story of the iceberg that got stuck, and the sea ice concentration at the beginning of the breeding season was higher, so they had to cross more. And there is also, but it's the results from that, they're not, I mean, to me they didn't seem very clear that they would gain any advantage at advancing their reproduction. There is a bit also of discussion regarding interspecific competition with the gentoos and the chinstraps in the peninsula regarding arrival on nest site. But yeah, in other regions, I don't think it plays such a major role for them, the phenology, as it does for other species such as king penguins, for example.
Yeah, I'm sure you're right. But does laying date vary? Maybe it doesn't matter too much when they arrive, it's whether or not laying date changes that's important.
Yeah, that's well, I don't remember, I'm quite sure I read about this, but what I can tell you from our data is that this advance that they would take on the arrival at the colony was completely offset by the laying date. The laying dates were almost always the same. So they do this buffering in between the arrival and the laying. So maybe, but this would be interesting to see how this translates into their breeding activities, because what it means if they have shorter periods is that they have less time to build the nest and less time to feed the pair bonds. It's not the right term, but to bond the pairs.
Yeah, sure. Thanks. So, Téo, obviously there's a very strong focus on sea ice here, and I'm not going to actually talk too much about that because there's going to be some more questions on that from Emily. I was just wondering about other indirect effects of sea ice. I mean, is there, and the obvious one is, so sea ice is mostly we're talking about, bottom-up control, but is there any difference in the way that the predators, that the skuas might respond to the sea ice situation that could be maybe exacerbating or ameliorating effects on the penguins, depending on whether the skuas fail, for example, and that sort of thing? I mean, just essentially, what about predation and what role does that play?
Yeah, for the squaws, actually, I'm not sure. I know that they may have different strategy among individual squaw, that some forage at sea, some forage more on the penguin colonies. So maybe when there is a lot of ice, the squaws don't have access, so they shift their predation on penguins. I know there is a really nice paper on that by Lise Viola and Christophe Barbeau that I read, but I don't remember the conclusion. They modeled the dynamics of the two species together. Actually, Christophe did that in many papers. I don't remember the results, but the thing I can tell you about is not with squaw, another predator, the leopard seal, and in the Ross Sea, there is not much information about this, but they are, in Adelie Land, like, we rarely see them, the leopard seal. There is one or two for 40,000 breeding pairs, it's not a lot. But what they found in the Ross Sea is that the predation risk is quite high, and especially when the sea ice makes it that they have to use specific areas where the seal can hide. So probably it can play a bit also, but it's tough to quantify that, especially if it's farther away from the colonies.
Yeah, thanks. Okay, so again, we'll stick with life histories. You made a really interesting comment, in your introduction, and you were talking about the difficulty of distinguishing between life history strategies from life history trajectories. You put it very elegantly, and even then within life history trajectories from effectively the distinguishing cause and consequence from intrinsic adult quality. And I couldn't agree more, but how do we really tell what are strategies to some extent within individuals, and how distinguish that from effectively individuals just reacting to their immediate circumstances and their recent breeding outcome?
Yeah, I scratch my head on that question a lot. And the only answer I came up with, I have no idea if it's valid, but I would frame it as a fitness equivalence. So if you don't have fitness equivalence, you have trajectories. If you have fitness equivalence between different trajectories, they become strategies. So I would really bring it to this idea of fitness equivalence, but it's not really practical to apply it in nature to actually see that. Yeah, but hopefully, one thing I read about recently, that you may not going to like the answer, is that actually for population dynamics, it may not matter this much. I thought before reading more for writing the thesis, I thought this was potentially very important, and we have paper by Stéphanie Genevrier, for example, showing that it may be important, but recently I read another one on snakes, or actually reptile, I don't know which one, I don't remember which one, saying that they modeled different scenarios of individual heterogeneity in strategies, and the impact, or turtles, maybe it was turtles, the impact wasn't this big. But yeah, sorry, I slided a bit.
No, that's great. What have we got? Oh yeah, this is in wrong order, but anyway. So do you think obviously there's problems for Adelie penguins if there's changes to sea ice extent. And of course, we were seabird ecologists, we consider across species comparisons. Do you think the future of some seabirds might be more secured by, stepwise shifts in range, like the breeding range? So effectively that would be something akin to your black swan events, but that suddenly subantarctic species might start breeding in the Antarctic. And first of all, might that happen? And second of all, for Adelie penguins, is there anywhere else to go? Or effectively, for the Adelies and emperors, really, if they can't cope with these sea ice conditions, that's it. They're never going to. Would they ever survive in a subantarctic environment, or is that really not an option? Is everything basically become more polar, and then options disappear?
So yeah, regarding your first question, I think definitely some species can maintain themselves and can benefit from a shift in distribution, especially, as you said, the subpolar species coming to more and more to polar areas. This may, just the little information I wanted to add, this may depend on whether the new habitat, the newly available habitat remains favorable all year long, if they are not migratory, of course. And this is important. I think this is also highlighted with Adelie penguins, that the environment at different timescale may have different effect. So for those species to benefit, they would have to have a favorable habitat all year round. And now on to the second question. They can actually go south still a little bit, at least I think for the 21st century, especially in the Ross Sea and the Weddell Sea. I think the Weddell Sea in the Ross Sea it's more, they have more papers by Ainley, for example, as early as 2010. I mean, it's not so early, but anyway. And in the Weddell Sea, what we are seeing now and realizing is that the Adelie population on the peninsula that are Weddell Seaside, they are doing great, actually. You have colonies that Céline sampled 2 years ago that are above 1 million breeding pairs. So it's quite huge. And the emperor penguins, then, yeah, as you say, we will never see them in the subantarctic, that's for sure, because no breeding platforms. But I think for them, the population, they may be able to use the Weddell Sea more and more as this sea become less icy. Because at the moment, the problem with the Weddell Sea is not that there is not enough ice, it's that there is too much. So yeah, but this will work obviously only for a time, and also the idea that is important is that it may likely not be able to host big enough population to ensure that they are not wiped out by such extreme events, for example.
Yeah, thank you. No, good points. Okay, and so back onto methodological ones. Did you consider using the other kind of IPM, so integrated population models, so you could effectively analyze your capture mark recapture data, but then inform that with the count data? Did you think about that, or?
Yeah, I thought about it, and it was kind of a trade-off, because I mean, the way I see it, well, maybe I shouldn't say that, but the reason I was drawn to all this is not for the math, it's for the biology, the ecology. So when I look at these models, I think the outputs, what we can get from them is super interesting, but the math maybe, I mean, they frighten me a little bit. But the reason is what usually the way I see it is that they are used when it's really difficult to estimate the vital rates, because you have sparse data, because your data isn't clear, maybe your recapture rates are very low. That's not the case for us. So we were already able to have quite a good idea on the vital rates we were interested in with the usual capture recapture models. And also something I'd like to add is those models, they're great. But if you have more than one vital rate that is missing and you try to estimate it, and you also have papers saying, yeah, they do nonsense. They just like find the average value that maximizes the likelihood or whatever optimization function you're using, and yeah, here you have it. So I think it's super useful in some situations, but in ours where we were already able to have quite good vital rates data with the classical models, it's yeah, maybe not needed so much.
I think you're right. Great answer. I must tell all the people I know who work with integrated population models and remind them of exactly that. No, I do tend to agree. You know, in a data-rich situation, they're perhaps not necessary. Yes, go ahead.
But yeah, sorry, it's just that it's funny. I see Guy laughing because we talked about this a lot this week. Another thing I didn't tell much about is here we consider that population variation in size, they are driven by vital rates. But there is actually, for population that are in what we call a transient state, so that are not at the stable age distribution, they may also be driven by change in the age structure. And I think this is one of also the big, something that makes the IPM, the integral, not the integrated population model really useful. To be able to assess also this age structure that may have some influence on the dynamics.
Yeah, thanks. And so just a general comment. Are you certain there's no transponder loss?
I prepared a slide on this. No, that's my, yeah, but yeah. But we could test it actually, and that's also a discussion we had with the collaborators. I'm sure Céline thinks about it every once in a while. No, we don't. That's unfortunate. I mean, sorry, I don't. Maybe other people know. But yeah, I quoted this website. There is a journal called RFID Journal. I was quite surprised. And apparently the kind of founder and editor of the journal said, no, I don't think they, so. I mean, it's not super hard evidence, but...
yeah, I mean, I think the one issue is the malfunction in the RFID tags. The other possibility is they're lost shortly after implantation. So I wonder whether that's more of an issue than people tend to suggest, which of course you could test it by injecting a couple of pit tags into the same animals. It wouldn't be that hard. Or unless do they interfere when you're going over the detector? If you have two RFID tags.
Yeah, that's bad. That it doesn't work. And we actually have the problem because the way we do the pit tagging of the cohort, so we go in the colony, we catch the penguin, implant the tag, release it basically, and we put a bit of green paint on the cheek. But sometimes when the first one we do in the season, we try to do it really condensed, but if there are storms and stuff like that, it's a bit delayed. We may, usually we check if it's not already implanted, but we have some birds that have two pit tags, and what happens when they cross the weighbridge is that sometimes it's one, sometimes it's the other, and sometimes it's not at all. because there are interferences between the radio signals. But I have, with Coralie, that's funnily enough, that's here today, I have dissected juveniles that died after being implanted, not because of the implantation, just because of the storm or predation. And I mean, you could see that the pit tag was quite well in place. Well, it's worth what it's worth as information. But so yeah, I would say we go, let's say 3 centimeters, the pit tag is deposited 3 centimeters below the hole, and also usually when the penguin is standing up, the hole is towards the ground, and the pit tag is towards the ground, and the hole is up, so the chances that it comes out are lower.
Yeah, no, that's good. Okay, so I might just follow up with if we've got time, yes. I'm just going to follow up on something that Sandra was asking about, which was about whether you have planned or thought of testing for the effects of trip duration in incubation. You know, I mean, you could give good reasons why this focus on brood guard is important, but it might also reveal something about the importance of sea ice extent at that time of year. I mean, because of course some animals do fail at hatching, and you could also, if you could identify hatching birds that have failed to hatch or not, that you would still glean some useful information from repeating the analyses in incubation. If so, do you think that would that show anything different about sea ice? About the benefits of sea ice, or what the optimal sea ice conditions are if you were to repeat the analysis depending on incubation and analysing incubation foraging trips?
Yeah, I would say this really depends on how much does sea ice block success to predictive foraging rounds. And in the case of incubation, the site they go the most during this period, at least in Adelie Land and in other regions as well, is the shelf break. So the moment where the bathymetry really goes down, and because of the upwelling, this is a productive zone, so they can go there and target so it's the Antarctic krill that they find there, opposed to the crystal krill that they get in the ice. So yeah, apart from that, I don't think, maybe too extensive. Again, if it's really, really too extensive and they have to walk too much, it may lengthen the incubation trips, and then it may be worse for reproductive success, but yeah.
Actually, how much do you know about diet?
Yeah, that's
how much whether it might have changed or might change depending on environmental conditions?
How it might change this, I'm not... The paper on the sea ice trend, the summer sea ice trend, maybe we can put it again. I mean, for now it's a preprint, and they quantify the time of emergence of climate change signal. Oh, it's not looking good now, but also for this figure for sea ice, penguin, and fish. And I think I honestly don't remember the direction for fish and krill, but what they are seeing for Adelie Land, I'm not sure, but what they are seeing in the Antarctic Peninsula is that the fish, the ice fish, the Pleurogramma, that is associated with sea ice, its distribution is contracting southward. And what they found in their analysis there is that the breeding success the chick mass, so probably an index of their survival, is correlated with the abundance of fish in the diet. This is also true in the Ross Sea, So yeah, if the ice fish decreases, probably it's not going to be good for them. But at the same time, you have colonies where the breeding success is still high, in excess of 1, and they feed at 99.9% on Antarctic krill. So the importance of fish may really depend on the colony. But it's really tricky. I would really like to know why some can handle 99% krill, and why some they need fish in their diet to be able to breed. That's I wonder.
Yeah, it's a good, tricky question. Okay, another generic question. Do you think it's better to be, if you're a chick, in a cohort that's large, and therefore, because there are potentially some benefits in terms